lab on Kaposi's sarcoma-associated herpes virus, or KSHV, and in particular the protein ORF68. And so before I get started, I'd just like to thank the organizers for the chance to speak to you today, as well as all of you for your attention. So KSHV is a herpes virus, and herpes viruses have these large, complex virions where the double-stranded DNA genome is encapsulated by an icosahedral capsid, which itself is encased in this proteinaceous tegument, which is then, again, further enclosed by a lipid envelope, which is studded with viral glycoproteins, which you can see here in this HSV1 electron micrograph, where you can see the capsid and the lipid envelope, and in this cryo-EM structure, where you can actually see the really dense DNA of how tightly packaged this is, as well as the portal protein through which the genome is threaded, both during encapsulation and during infection in the cell. So with such a complicated virion, the process of making them is also pretty complicated. It's highly regulated and well orchestrated. So what happens is there's a head-to-tail concatemer formed during genome replication, where the genome is separated by terminal repeats. Once it's replicated, this viral complex called the terminase will bind it, bring it to new empty procapsids, which themselves are formed by the capsid proteins being formed onto a scaffolding protein, which is then degraded out of the middle. The terminase can then translocate this genome into the capsid, forcing out that digested scaffold, where it will continue to translocate it until the capsid is full. The terminase cuts the genome then in the terminal repeat, which is then released, and this can go on to package new capsids. And so this complicated process is conserved throughout all herpes viridae. And what was really interesting to us is that there are several proteins that are known to be required in all herpes viruses. But one protein, which is ORF68 in KSHV, is known to be essential, but no one knew what it did. It didn't have any real defined functions other than it's required. So that's what was the basis of my PhD work so far. And so the first thing we did was to make a deletion mutant of ORF68, so KSHV that cannot express ORF68. And we wanted to look at this, its ability to package DNA, by doing a southern blot for the terminase. And the reason that's really useful is there are 20 to 40 copies of this terminase, which I've kind of shown here in blue, in the just one copy of the genome, which is circular when attached to the host chromatin, but when it's replicated, it's this head-to-tail concatemer, as I just said. And by looking to see if these terminal repeats are cleaved, you can actually really easily look if the virus is able to package its DNA. But so the way this assay goes is you have lytically replicating virus. It's either successfully cleaving and packaging the genome, or it is not successfully packaging it, and in that case, the terminal repeats are not cleaved. What we do is harvest that DNA. I digest away the host genome as well as the viral genome with a restriction enzyme. This is not cut in the terminal repeats. And then just by running it out on a gel, you should see a ladder of terminal repeats, because while the exact area, or the exact site of cleavage in the terminal repeat is conserved, the amount of terminal repeats on either end that are left is somewhat random. So basically, we should see a ladder if the cleavage is successful, separated by around 800 base pairs. And so in wild-type cells, what we do see is this ladder of cleavage events, and additionally, you see this replication of the genome from the latent stage to the lytic stage. But without ORF68, you don't see this laddering. You do see an increase in genome number, which we've also confirmed by qPCR, showing that the virus can replicate its genome, but it just can't cleave it here. And then when we repair that mutation, it's totally fine again for cleaving. So this made us think, is packaging not happening because there are no intact capsids, or because there's something wrong with the actual packaging process? So to look at that, what we did was transmission electron microscopy, and here you can see the virus of a cell infected with wild-type KSHV, and you can pretty easily see these capsids. Those are divided into A, B, and C capsids. C capsids have this genome. This is the DNA. These will go on to be infectious virions. And then there are also B capsids, which, if you can see, have this little ring, which is actually the cleaved scaffolding protein. So these either just haven't encountered the terminase and DNA packaging machinery, or there is some problem in doing DNA packaging. And then A capsids are actually just empty icosahedrons, which are formed by the genome attempting to be packaged and forcing out this scaffold, but then falling out, or there's some other problem. And in ORF68 SNP cells, what we see are only B capsids, which tell us that there's no problem in making capsids, but there is a problem in packaging the DNA. And so this kind of led us to hypothesize that ORF68 might be actually interacting with the genome itself and in some way either bringing it to capsids or processing it in some way. And so what we did is we purified ORF68 recombinantly from 293 T cells and did a EMSA. And what you can see here, this is one subunit of the terminal repeat. And as you add ORF68, you get this shift up of the terminal repeat, showing that ORF68 can bind it. And you see higher order structures, which are probably just multiple binding events. And then when this is quantified, you see that the affinity is around 50 nanomolar, which is pretty robust and is very similar to other proteins that we know to bind this same region. So something else we saw while we were doing these assays was that inclusion of metal cations, specifically magnesium, led to not only loss of shifting, this is the same EMSA probe I was just showing you, loss of shifting, but now degradation of the probe down to, as you add more 68, down to this small fragment. And that is totally inhibited by the inclusion of EDTA, which would chelate the magnesium. And then looking at this further, we can see that not only can it use magnesium, but it can also use manganese and to some extent cobalt and nickel, though calcium and zinc are not sufficient. And so this is all pretty consistent with metal-dependent nucleases, which we think ORF68 may be. So basically at this point, we think that ORF68 is binding to the genome, maybe processing it catalytically, which is why it's required for this DNA packaging event. But in the Glaunsinger lab, we had also produced something called the KSHV interactome in collaboration with the Krogan lab. And what that was a series of host protein, viral protein interactions identified by mass spec. And the major complex that came out with ORF68 was actually the proteasome, which is the major ATP-dependent protease present in mammalian cells. It degrades most of the protein that's made, and it's involved in many cellular processes. And furthermore, ORF68, as well as the orthologs and other related herpes viruses, has something called a gate-opening motif, which is on the very C terminus of the protein. It's this hydrophobic tyrosine any amino acid. And this was really exciting for us because this is a motif that proteasome activating proteins have which enable them to access the actual proteolytic machinery of the proteasome. And so much better than I could ever explain it is this animation by Janet Wassa, where you just want to concentrate on this 20S core here, which is the actual chambered peptidase. And so the peptidase domains are here shielded in the middle so they don't just degrade all the proteins. And they are, access to them is managed by this 20S gate. And when the gate opening motif sits down in those inner subunit pockets, you get this opening. So that's why this gate opening motif is such an important motif for proteasome interacting proteins. But to make sure all that was actually happening, the first thing I did was just transfect in to 293 T cells, a tagged subunit of the proteasome, so I can pull out the 20S, the proteolytic part of the proteasome. And I looked for ORF68. And here you can see ORF68 does co-purify with it through transfection. And then this is also true in infection. This is purified a slightly different way. But in either case, we see ORF68 associated with the 20S proteasome. But just because it's there doesn't mean it's doing anything. So next, we looked at the ability of the proteasome of ORF68 to activate this by something called a GFP half-life assay, where we watch the degradation of a model substrate in cells. And so here you can actually see that wild-type ORF68 didn't seem to do anything, which was, this slope is just the rate of the substrate going away. But what we did see was deletion of the gate opening motif or blocking it with a C-terminal tag decreased the rate of GFP going away, which meant that the proteasome was less active on this substrate. And the difference between these two is mostly, we think, explained by this LYA mutation being somewhat destabilizing. So if this was a little more stable, it probably would have a similarly shallow slope. So this at least suggested to us that ORF68 could interact with the proteasome. It actually had a functioning gate open motif. And just to add to that, something we noticed while purifying the ORF68 was that it purified as an oligomer. And not only that it purified as an oligomer, but by doing some negative stain EM and recently some cryo-EM, that ORF68 is actually a pentameric ring, where you can see there's these 5 subunits and there's actually a central pore, which is very similar to a lot of cellular proteasome activators, where they sit on top of this chambered peptidase, can open them and translocate substrates through them. We don't have any evidence for this actually translocating anything, and hopefully pretty soon we'll have an even higher res structure of this. But being good virologists, we wanted to make sure that all of this was actually important to the virus. And the way we do this is by just a supernatant transfer assay, assaying how much new virus any of these mutants we have make. So in this assay, wild-type virus makes enough virus to infect about 100% of our target cells. Deletion of ORF68 here totally ablates the production of new virions, which is in agreement with past work. And really excitingly for us, deletion of just these 3 amino acids off the C-terminus of ORF68 out of the entire KSHV genome is enough to totally ablate the production of new virions. And then repairing that mutation rescues it back to 100%. So at this point, what we think is happening during DNA packaging is that ORF68 is binding to the DNA genome, somehow processing it possibly catalytically, allowing it to be properly packaged into nascent capsids. And then it can also bind to the 20S proteasome, maybe with other host proteins or other viral proteins, to form an alternative proteasome which may degrade different substrates, which may be useful for processing proteins off of the genome during packaging, or may have a totally different life in the cytoplasm of the cell. And so with that, I would like to thank everyone in my lab, my thesis committee, our collaborators in the Martin lab and the Kosker lab, as well as people who aren't in the lab anymore, and I would love to take any questions.
Questions? I'll start with one. So do you have any evidence that the ORF68 would associate with the terminase? Have you found it associating at that end, or have you looked at that?
So I've done co-IP experiments, and I've seen it to co-IP with some of these subunits, but it's not been found to be associated with the actual virion, which was something kind of, again, confusing that it was required but not associated. So I have some preliminary evidence that it may be associated with it, but we're not sure if that is where it's associating or not.
Okay.
Just wondering how you kind of parse out between 2 different models you have going on. Like, have you been able to mutate the nuclease activity?
No. So ideally, the really horrible thing with at least this protein is, or a lot of viral proteins, particularly herpes viruses, is that there's no, at least in any programs we use, they don't really predict them to have any structure. And so definitely nothing that looks like an active site, but there's no kind of structural data other than this, and maybe with our cryo-EM and hopefully some crystallography, we could find something that looked like an active site. There are conserved residues that are canonical metal coordinating ones that we could mutate, and ideally that would be a great way to separate these two to see if they're at all involved, yeah.
And are these pentameric rings related to DNA diffusion?
No, they are just, when they're just isolated, totally absent of DNA. These structures are just absent of DNA. I actually can't really find a way other than denaturing them of breaking the pentamer. No matter how much salt I add or how much detergent, they seem to really want to stay as a pentamer. So.
Okay.
Thank you.